Hi, everybody. Thank you very much for joining today. My name is Nicole, and on behalf of Megan, I'd like to give you a warm welcome to this Megan Masterclass with our guest speakers, Ole Jensen and Oscar Ferrante. We have a few housekeeping rules to go over. Please use the Q&A button on your screen to send in your questions at any time during the masterclass. There will be time at the end to answer questions, and any questions unanswered, we will try to reply in writing after the session. Please remember that this webinar is being recorded with the permission of our speaker and will be available on demand. And finally, please adhere to the Megan Code of Conduct and engage respectfully online. So I'm handing over now to our guests, and thank you very much for joining today. Hi.
Thank you very much, Nicole, and thanks to the Megan Masterclass for giving us this opportunity to talk about our FLUX pipeline for data analysis. So just by words of introduction, this pipeline is now published in NeuroImage, so you can get to the paper there and read more about it. We are a whole team of people that have been involved in developing this. So in particular, there's Oscar and Ling, who has been part of developing the NME Python part, and Thomas Menerik has been important for developing the field trip part. So this FLUX pipeline came about through a collaboration in the context of the Computation consortium. So this is a consortium based on an adversarial collaboration where we are collecting data here in Birmingham, and as well as a team of Wanlu collecting data at Peking University. And what we of course need for that collaboration is a common pipeline. So out of that pipeline came the FLUX that we're going to talk more about here today. And then also people in my group here, the Neural Oscillations group at the CHBH have contributed to this. So what is the sort of core background for developing the pipeline? In the sense that there's a lot of possibilities out there on the internet when you browse around for MEG data analysis. But so one of the key motivations is that if you are involved in multi-site studies, some sort of standardization for analysis is needed, right? For instance, in the Cogitate collaboration, it became very fast clear that we do need to sort of join efforts in order to define what we are doing, so we have similar analysis pipelines across sites. And of course, there's a lot of collaboration sort of growing on MEG data analysis, where different groups are involved, and for that we need a more rigorous approach to the data analysis. Furthermore, there is the training. There's quite a few people coming into the field. They need to be trained. Of course, if there's a jungle out there in terms of analysis approaches, it's very difficult to enter the field. But if you have a more tight pipeline, that will also enter that effort. And then very importantly, there's the issue of reproducible science. You probably all know about the replication crisis. And of course, part of that is that we don't have sort of standardized ways of doing the basic steps in, for instance, data analysis. And again, by having a pipeline that we here propose should also help that issue. Another point which is becoming very pertinent is the notion of democratizing science. So there's a lot of talented people out there in research. Not all these people would have access to, say, MEG data because they don't have an MEG system at their site. However, there's more and more data being shared, and these people can then download those data and contribute very constructively to the research. However, we also want to make it easier for those who want to partake in getting into the field, and again, having a pipeline like this would facilitate that. Okay, so to be more specific, for some of the challenges that we have faced when doing MEG analysis. So there's a lot of excellent toolboxes out there for doing the analysis. So of course, examples are Field Trip, NME, Python, Brainstorm. what toolbox should we use, right? So our idea is that, well, I mean, if we provide scripts that can be used for multiple toolboxes, maybe that is then becoming less of concern. Furthermore, if you have selected a toolbox, another problem is that there's many options you can take. Even if you go to NME Python and want to do ocular artifact reduction, you can do different approaches and they might be equally good. So why not just settle on one approach that then can be used as a standard? Okay, so how is the FLUX pipeline then set up? Well, first of all, there's the neuroimage paper I pointed you to, so that sort of provides an overview of what the pipeline is doing. More importantly, we also have specific steps and parameters defined, and you can look those up in the documentation, which is also on the internet on GitHub. So basically, what we provide is scripts and parameters implemented in Jupyter scripts for NME Python and live editor for Field Trip. And Oscar, you will hear from in a minute, would show you some very specific examples. What we also do is that we are providing suggestions for text that can be used in manuscripts and pre-registrations. And furthermore, we also provide a data set that can be used for training and education. So when you go to the website for the FLUX pipeline, what you will find is some short descriptions, and then here is some chapters for the NME Python part of the FLUX pipeline, and it will then take you to the different descriptions of how to do different parts of the processing of the data, and it will take you all the way from the data set itself and then further on to doing the pre-processing, some time frequency analysis potentially, and then the source modeling. So the flow is also illustrated as you see here, where you first read in the data and then you can identify bad sensors. There would be pointers to how to do the noise reduction using various kind of algorithms. That could be SSS or the third order gradient for the CTF system. For NME Python, there's a very convenient artifact annotation that allows you to mark artifacts that then can be rejected later when doing the trial segmentation. And there's also the ICA artifact reduction. So after the trial segmentation, different steps can be taken. That can be calculating event-related fields, the spectral analysis using time frequency representations, or multivariate analysis. And after that, one can do source modeling to, for instance, localize where brain a given effect in the frequency domain is occurring. So when you go into one of the chapters, in this case for NME Python part of the FLUX pipeline, it will take you to GitHub, and what you then find there is these scripts, and they are implemented as Jupyter notebooks. And these Jupyter notebooks are very convenient because they allow you to or allow us to make some documentation for the code, and furthermore it's possible to run the code itself within the Jupyter notebook. And furthermore, the output of the code, the graphics, will also be shown directly in the notebooks. So this is just a very convenient setup for how to document documenting the pipeline, but also for using it in education. Furthermore, what we provide at the end of each chapter is some suggestions that can be used for publications or pre-registrations. So you have probably noticed when you read papers from different groups, there's a big difference in what people report. And then we also want to sort of to tighten that up by providing concrete text that people simply can copy paste into the papers. And we have tried our best to sort of shave down to the core parameters. What you also can find in the chapters in different parts of the analysis steps are questions. So that should make it easier to integrate these chapters into education, right? So personally, I'm running a course, and I have the FLUX pipeline as part of the practicals for that course. And then we are using the questions from the pipeline to be used in the education and for the homework. So as I also mentioned, we are providing an example data set for the pipeline. So it's a simple spatial attention paradigm that people are asked to attend left or right. So after a second, there's some movement gradings coming on, and then there's a little task. So basically, we have chosen this paradigm because it produces strong modulations in the alpha band. The gradings induces strong gamma oscillations. Furthermore, there's the behavior with the button responses. And we also see robust event-related fields. So it's sort of a data set that allows us to do to get robust modulations of different kind of brain activity. So Oscar is going to show you some more details, but I'll just show you a few examples. So here's an example for where we are using multi-tapers to characterize the gamma band activity. So the multi-tapers was introduced to MEG analysis by Pascal Fries back in the days, and they allow for controlling the smoothing in the spectral domain. And what you should note here is that what we see with field trip here to the right, we can also see in the analysis from NME Python, the color maps are different, but basically we can find exactly the same results. It also holds for source modeling. So here's an example of using the Dix source modeling approach that was introduced by Joachim Groß and is very convenient for localizing oscillatory activity in the frequency domain. And what you should note here is that we can localize the gamma band activity to early visual cortex. And again, we get very comparable results to when using field trip and NME Python. There's a little side issue here I want to bring up. There's been some confusion on how to deal with the rank deficiency introduced by the SSS noise reduction algorithm used in the MEG system. So basically, you start with data from 306 sensors, and after using the noise reduction, the rank of the data is reduced to about 70. So what to do about that? Several solutions has been proposed. However, what we propose is to use the truncated pseudo inverse. There's also something you can read more about in the paper. So, but basically, when doing the pseudo inverse, it's the first k diagonal elements being used. So it also has the advantage that no further regularization needs to be used here when doing the source modeling. So to make a long story short, I think we have a robust solution which already is implemented in MEG Python, but we provide the guidance for how to do that more precisely. So we have a good solution for how to deal with this issue of the data being rank deficient after using SSS. Something we also have introduced is a chapter on how to do multivariate pattern analysis. So as you're probably all aware, multivariate analysis is becoming more and more popular and used in our fields. So it's based on machine learning algorithms, and NME Python is integrated with the scikit-learn toolbox that provides a lot of different powerful tools for doing classification in this case. So here's an example where we can classify people attending left versus right, but of course this can be applied to a lot of different cases. So what toolbox to use? There's a lot of toolboxes out there. So I should just mention here that what we have found when comparing NME Python and field trip, I should mention we used to use field trip, but we are switching to NME Python. But basically, what can be done in field trip can also be done in NME Python. I should also mention there are other toolboxes like Brainstorm. So recently there was a comparison done by Amit Jaiswal, and it's published in NeuroImage 2020, and they found that Brainstorm actually was very powerful compared to the other toolboxes when doing source modeling. I mentioned that we are now switching to NME Python, and the reason being that we would like to sort of future-proof our future research, in the sense that one of the advantages of Python is that it is free. As I mentioned before, there's also the element of democratizing science and research, and of course, when using free tools, more people can join the game. Furthermore, at least here at our university, Python is being taught as a basic programming language, so it's easier for us to introduce the tools, the NME Python and FLUX pipeline to these students. Furthermore, as I mentioned before, NME Python are making use of the scikit learn toolbox, so it's a very powerful machine learning toolbox, allowing us to use multivariate analysis. And furthermore, there's also a powerful stats model module that sort of is replicating many of the statistical functions in R. But nevertheless, of course, it's a personal preference what toolbox to choose, also depending on what programming language you're most comfortable with. So a few words on the future directions. What we want to do in the FLUX pipeline is, of course, not to keep it static. It needs to be continuously developed. What we want to include is the MEG bits format. We should have done it, we just ran out of time, but that's the next priority for the FLUX pipeline. There's a lot of excitement on OPMs. So what we also want to do is to make the pipeline more suitable for OPM data analysis. Furthermore, we need to expand on the pipeline for group analysis. We also want to include tools on hidden Markov models, which have become very popular for resting state analysis. Furthermore, rapid frequency tagging is something we do in our lab, and we have a chapter on that coming up. And finally, we also want to make some courses, both online and in person, for how to use the pipeline. I just recommend you to try it out and have a look at the website. And now I will switch to Oscar, who's going to give you some more specific demos on how to use the pipeline.
Thanks, Ole. Okay, so as Ole said, I'm going to show you our pipeline. So for a matter of time, I wouldn't be able to show you every single step, but we decided to focus on what we think are the most important things. But feel free to go to our website and look at all the materials that we put there. So this is the website, and as Ole mentioned before, we have a paper about it. So if you want to know more theoretical stuff about the analysis and why we propose some methods instead of others, there are a lot of information in the paper. Then in the website, we also have a nice video that Ole made. And there is also a standard operation procedure document that is a document in which we show how we collect data here in Birmingham with the MEG system. So we explain step by step how we collect data, and we think this is also something that could be useful for running experiment in other places. Of course, it needs to be adjusted to your equipment, but it's also here available on the website. But let's go to the meaty part. So here you see the FLUX pipeline for MNE Python with all the chapters, and here you can see the same chapters, this time with field trip. So there are some small differences between the two pipelines, but the results that we obtained with both are quite similar. For a matter of time, I will focus on the MNE Python part. First, I want to tell you how to use the MNE Python FLUX pipeline. So here you see how to install it in the introduction part. So what is important is to first install Python on your computer, and we recommend to use the Anaconda software that you can find on the Anaconda website. Just download it, and then when you install that software, you will have Python running on your computer, as well as some tools that are very useful, like the Jupyter Notebook that we use here for the pipeline scripts. Then after installing Python, you also have to install MNE Python. And if you go to the MNE Python website, it's a fantastic website. There are many resources. And here there is instruction on how to install MNE Python. They recently released a standalone installer, but so if you're not used with coding, you can also install MNE Python and Python using this installer. So what we recommend in our website is to use the conda command. It's just a couple of lines of code, so it's also not too complicated. Now you have Python and MNE Python ready. Then what you need is data that we are using in our pipeline. So the sample data can be found following the link on our website, and it's shared here on Figshare, and you can just download the files from here. So there are 2 files with MEG data and other 2 files that are used for the SSN Maxwell filtering step. Now you have the data, you only need to download the codes. So all the codes are on our website. You can just go to the website, copy and paste the part of code that you need. But if you want the full pack, you can just go here in our GitHub repository. If you go to the main page, you click on code, you can download a zip file with all the codes for both the field trip, MNE pipeline, the SOP, and everything. Okay, now we are ready to start. So first, sorry, I won't be able to walk you through all the steps. So we highly recommend you to go to our website and explore it yourself, and also try it to your laptop. So after installing everything, so what we recommend you do is to first have a look at the data to see if the data are good. Then start removing artifact with the MEG system. We recommend using max filter and the SSS method. Then the artifact annotation that Ole mentioned in the introduction is also done to take care of artifact generated by movements or muscle contractions, as well as breaks during the recording session. Then we recommend to start the ICA decomposition here to remove artifacts related to ocular and cardiac components. And I want to walk you through this part because I think it's quite useful to see what we recommend to do. So the ICA decomposition is the independent component analysis decomposition. That is one method, is not the only method, but it's one method that is commonly used to reject ocular and cardiac artifacts. And it's performed in 2 steps. During the first step, you extract the components from your data, all the independent components. And then in a second step, after identifying those components related to artifacts, you have to project the components out from the original data. So how do you do it in Python? So here we start the first step. Yeah, don't be scared about all this code. It's quite simple what we're doing. We are just looping through the 2 MEG recordings that we have. Then we read the raw data here with this line of code. And then this is an important part, because what we recommend to do is to first here we create a copy of the raw data. Then we downsample the data to 200 Hertz. So this data set was recorded with an original sampling rate of 1000 Hertz. And then we recommend to bandpass filter between 1 and 40 Hertz. So the reason for downsampling the data is because the ICA decomposition takes time, and it's a computation quite heavy, and it's better and it works better if you have smaller data sets. And also you can downsample the data because here we recommend to bandpass filter the data. So the 40 Hertz low pass filter is here because the components that we are looking for are known to be slower than 40 Hertz. And we also recommend to use a 1 Hertz high pass filter to remove the slow drift that can have a bad impact on the ICA decomposition. So now that your downsampled and filter data are ready, you can start the ICA decomposition, and with MNE-Python, you first have to set some parameters, so to decide what method to use. So we recommend to use the FastICA method, but there are many other methods that are used. So we recommend FastICA because it's well established and widely used. Then another important parameter to set is the number of components. Here we set it to 30 because usually you don't get more relevant components than 30, but you can also have more. So the maximum number of components is the same number as the number of channels that you have. So don't pretend to have too many components. So and then after setting the parameters, the ICA decomposition is run on the data, on the resample data. And here you can see the output in this table, and we see that the number of components is actually 30, and that the explained variance that these 30 components explain is 95%. So it's important that the components are explained a very high percentage of the data, the variance in the data. Now we have all the independent components, so what we can do is to use this plot source function to show the time course of each single component. And then here, is it possible to use algorithm that automatically identify the bad components, but we also because this is meant to be used for educational, so we recommend to use to do it also visually, so they also to know what you are rejecting. So for instance, here an example is that the first component 000 shows a pattern of that activity related to blinks with this response here that are periodic. There is another component, it was component 14 that I want to show you, and this is a typical the time course that you see related to saccades. So with this quadratic change, like one that I'm showing you. So now we have the blink, saccades, so you can also find other components. Then when you have identified all the components, it's also good to look at their topography using the plot component function. So here, for instance, we can see that component 1 is indeed related to high movements because the topology is very front end, and it's the typical topology that you have with MEG data related to blinks, and 14 is the typical topology that you see with saccades. So if you work with EEG, probably you have noticed that the pattern is reversed, just because here we're looking at magnetic fields, which is the opposite, has the opposite orientation than the current that you measure with EEG. Okay, then all the components are identified, so we just have to add the components that we want to reject to the exclude field of our ICA. And then this is an important step that I want to highlight, is that we are reading again the raw data, because once we are done with the ICA decomposition, we forget about the downsampled and filtered data, because the matrix that we obtain, the special filter, can be applied to any kind of data. So given that here the data are the same, so we can go back to the original data, keeping all the frequencies, and then with this function ICA apply, we are rejecting the component that we want to exclude. When you're done with the ICA decomposition, it's good practice to check the result. So what we do here is to plot a sample of the data before the ICA decomposition, showing this this ocular related artifact, and then showing the same piece of data this time after ICA decomposition, and you can see clearly that the artifact is gone. And as I said, at the end of each chapter, you see an example of what to write in a pre-registration or publication in the meta section. Here you see everything that we suggest to write in the ICA part. Okay, that's all for the ICA. After the ICA decomposition, we can then extract our condition from the triggers and then epoch the data into trials. And then we're done with the processing. I won't be showing this part, but I want to show you some of the analysis. So here we have tutorials for event-related fields, time frequency representation, multivariate analysis for classification, and also source modeling. So I want to talk about the time frequency representation for a few minutes, because it's what we mainly do in our lab, and it's we have good recommendation on how to perform this analysis. So let me go here. So what we recommend to do is to treat low and high frequencies differently, because it's known from the literature now that low frequencies like alpha, they have a fairly narrow band that's about 2-4 hertz, while higher frequencies like gamma band activity has a way broader band. For instance, gamma could be between 60 and 90 hertz. And so because of these differences, here we suggest you to use two different approaches for the analysis. So the difference is just in 3 parameters that we are highlight here in this part. So the first parameter is, of course, the frequency of interest. That in this case, we are interested in low frequency, so it starts from 2 to 30 Hz in a step of 1 Hertz. Then what is important to set is the number of cycles. This will determine the size of the sliding window on which the Fourier transform will be performed. So for low frequencies, we need longer time windows. So here we set the number of cycles equal to the frequency divided by 2, which, if you do the computation, will give you a time window of 500 milliseconds. So for each frequency, you will have 500 milliseconds of sliding window. Then the third parameter is the time bandwidth, and this will influence the frequency smoothing. So here for the low frequency, we set this is the time bandwidth to 2 seconds, which, because we are using the multi-taper method, will give us a number of taper equal to time bandwidth minus 1. This means that for the low frequency, we are only using 1 taper. And this will end up with creating a spectral smoothing of around 3 Hertz. So you see here at the beginning the 3 parameters. Then in this example here, we are running this time frequency analysis on 2 different conditions, one in which participant has to pay attention on the left side of the screen, and another condition in which participant has to pay attention on the right side of the screen. Then after running the analysis, we can show the results using the simple function plot. This is for the left condition. And here we see the raw power. And you can clearly see here, for instance, that there is a high alpha activity before the time 0, which corresponds to the appearance of the stimulus, and then alpha decreases and almost disappears at this time point. So when you do your analysis, what your interest is what happens after the stimulus presentation usually. So for this reason, you have to use a baseline time window and correct your data for that baseline. So in this case, the baseline is before the time before the stimulus presentation, and we have some recommendation for the baseline correction. So we recommend to use the relative baseline correction, which correspond to the power for a given time and frequency minus the average power for the same frequency divided by the baseline time window divided by the average power during the baseline. And this will give you a measure that it's easy to interpret. And also in terms of where to put the baseline time window. So here, given that we are using a sliding window of 500 milliseconds, we recommend in this case to put the baseline in a time range, for instance, between 500 milliseconds and 200 and minus 500 to minus 250 milliseconds. In this way, we will ensure that the baseline will not overlap with everything that is above time 0. So I was saying that this is easy to interpret, the relative baseline, because if you have, for instance, a value of 0.2, this means that the power in that time point is 20% higher compared to the baseline. So it's also easy to interpret in that sense. And here we look at the same data as before, but this time with the baseline correction, and you can clearly see that there is a depression here in alpha band activity. This is also, then we also show how to show the topo map of this activity, like here in this plot here, we see clearly that it's localized over posterior sensors. And this is what happens, how we recommend to analyze and show the data for a single condition. But when you want to compare two conditions, so our recommendation is to not use a baseline correction, but instead to calculate the weighted difference between the two conditions. Here, for instance, we have attention left and attention right. So we can calculate the lateralization index, what we call here power lateralization index, which is equal to the power in the first condition, like attention right, minus the power in the second condition, in our case attention left, divided by the sum of the two. And this will give you a measure that is interpretable in the same way as the what I showed you for the relative baseline correction. It will tell you in percentage how much one condition is higher or smaller than the other. And here we can show the plot and see that indeed there is a lateralization effect with stronger alpha depression on the left hemifield. This is because the difference is right minus left compared to the other side of the brain. Okay, so but what about higher frequencies? So for higher frequencies, the analysis, so the recommendations are the same. The only thing that we recommend to do differently are how to set these three parameters that I presented at the beginning. So in terms of frequencies, so we go from 30 to 100 Hertz at step of 2. In terms of number of cycles, which determines again the width of the sliding window, so we can have a smaller sliding window because we have faster frequencies, meaning more cycles per second. So here we recommend to use a number of cycles equal to the frequency divided by 4, which gives you a time window of 250 milliseconds. And the time bandwidth, so we recommend for higher frequencies to use a time bandwidth of 4 seconds, which will give you a total of 3 tapers, and will result in a spectral smoothing of at least 6 Hertz. Everything is the same here. I just want to show you just one figure here, like you see the gamma activity between 80 and 90 after the CMOS presentation. And this is with the relative baseline correction. Okay. And also here we have at the end all the recommendation on what to put in the pre-registration or in the publication. Now I still have a few minutes, so I will quickly go over the source modeling part. So here we focus the source modeling on the oscillatory power. And in this context, we recommend to use a beamforming method, and specifically the dynamic imaging of coherent sources method that all I mentioned before. And in order to do the source modeling with beamformer, you need different ingredients. So the first ingredient that you need is the forward model, which is in a few words, is a matrix that allows you to go from the brain model to the sensors. For a matter of time, I cannot show you how we recommend to do it, but if you go to our website, there is a tutorial. We explain step by step how to create the formal model in both M&E and filtering. Then, of course, you need the data. That's another ingredient. And here, for instance, we will focus the example on the alpha power activity. And another ingredient that we need for the DICS beamforming are the cross-spectral density matrices. So these, in short, will tell how much information in terms of spectral activity are shared between sensors. And you can compute this using a simple function. Here we focus, as I said, in the alpha band, and you will look at the both baseline and in an active, so after stimulus presentation time window for both the left and the right attention conditions. But we also need this, we also need to create a combined matrix for the covariance metric that can explain the whole data set, because if you create these cross-spectral density matrices on a single condition, that everything that is happening in that condition will influence your source modeling, which is not something that you want to happen. Then the last ingredient is the computation of the rank. So Ole did a very good introduction of this and how it's done. So, and as Ole mentioned, so these the rank are reduced by the preprocessing. So, for instance, the max filter SSS method is reducing the rank from 306, that is the same number as the number of channels, to around 72. Then also the ICA decomposition does reduce the rank. So for each component that you reject, one rank will be removed. So that's why we end up with a total of 69. Now that we have all the ingredients, we can put them together. So I don't have time to explain everything here, but just imagine that what we're doing here is to create a filter that we can use to go from the sensor information to the brain information. So with the beamformer, what we do is to create a grid of points inside the head, and then estimate the power of each of these points. So given that there are so many points, because there are many more points than lectures, this needs to be estimated. And there are many methods to do this estimation. For instance, the ICS is one of those, and this is the well-known inverse problem. And this one solution is to use a method like the ICS and beamforming. So here we create the filter, and then once that you have the filter, you can apply this filter to all your conditions. Is what we do here. We apply the ICS, and then you have the source level data for each of your conditions. But there are some problems if you want to work with a single condition. So a way to solve some of the problem is to apply a baseline correction. And here, what we recommend is to use the same baseline correction that we used in the time frequency analysis. So here we're just collapsing the two conditions, the attention left and attention right. But what we do is the same. So we have the active condition minus the baseline condition divided by the baseline. And here we have the relative power change in a single condition. If you want to compare condition, again, as I said before, you just take one condition, you then compute the difference with the second condition, and you divide everything by the sum. So here, when you compare conditions, there is no need for a baseline correction. And finally, I can show you the result of the source modeling, just using the plot function. And here we can see where the alpha band suppression is localized in the brain. And we can see here that is in the parieto-occipital areas. Then we also have suggested about higher frequencies, and it's similar to what I said for the low frequencies. So what is important here is to change just a few parameters that will account for the spectral smoothing and the time window of the sliding window. And also here we have our recommendation for what to put in the paper. And I think that's it. Yeah, that's it. Now it's time for your questions.
Hi, I am joining in again to ask the first question, which is, does max filter and M&E's Maxwell filtering functionality produce identical outputs? Has anyone rigorously compared them?
Yeah, so I could answer that. So I had a chance to go through the questions while Oscar was speaking. So is it possible for me to send a message to everyone on? Do you know that?
Yes, here we go. I see yours. I'll put it to the entire audience. Okay.
Yes, so I just put in a link. Yeah, so on the NME Python side, there's a discussion on how using max filter as implemented by the MEG software versus NME software. So I should say that we have no reason to think that the NME software doesn't produce comparable or just as good results. There is the caveat though that the NME software is not approved for clinical use. That is the case for the MEG software. So if you're using your MEG system for clinical purposes, you might want to do the pre-processing in the MEG software with respect to SSS before then taking the analysis further. And it should also be mentioned that it's only recommended to use the max filter or SSS for MEG data. For instance, if you have a CTF system, you should probably use the third order gradient reduction. But I put in a link to the NME site where there's a discussion on this.
Okay, so next question is, shouldn't we be filtering before downsampling?
So that I also looked into, and so the downsampling algorithm in NME Python is doing filtering to avoid aliasing. So again, I'm going to put in a link for that as well. So there's also a discussion on the NME Python site. So yes, it is being done.
Okay, and the next question, 1 second. May I ask, how do you identify ICA024 being a component for Heartbeat's artifact? What features of it have been considered to be typical as Heartbeat artifact? Whether seeing the topo plot alone be enough to identify it as a Heartbeat artifact?
I can take this one. So maybe I said something wrong because that component was related to saccades, so to high movements, because it was, in the case, there was a clear peak, then a stable signal, then the signal was going back to the previous point, and that's indicated when the eye start moving and stop moving. But for the heartbeat, it's quite simple to recognize because you see this very rhythmic peaking signals, and also in terms of topography, you see the dipoles quite far from the head, like in lateral central areas around the head.
So I guess a related question someone has asked is, do you see the heartbeat components with MEG's system? I didn't see heartbeat in example ICA decomposition.
Yeah, that's because when we displayed the components, we didn't show all of them, but we removed one component that was in the next set of components that was related to heartbeat. And usually with the MEG system, I think it also depends on how noisy is the environment. For instance, here in Birmingham, we're lucky to have a very good place where we are recording our data. So we usually see in all participants a clear component related to the heartbeat.
Okay, next question is the FLUX pipeline planning on including some sort of automated ICA rejection?
Yeah,
maybe I can take this one too.
Yeah, yes, please.
Yeah, so yes. So the problem with the automatized ICA is, so it works quite simple, and with MEG there are already functions to do it. So if, for instance, you have you're recording the EOG signal, so you have two sensors just recording the EOG signal, you can just then compute the ICA decomposition, have all your single components time course, and then correlate them with the recording from the EOG. And then you can exclude, for instance, the two components that show the highest correlation. And the same, it can be done for the heartbeat by using the ECG. So in this case, we didn't do it because for the dataset, the sample dataset, we don't have this information. But yes, it's possible to do it.
Yeah, so also just to comment on this, there's always sort of this discussion on how automatic do you want the pipeline to be as compared to how much sort of visual inspection does it require. So here there's also the educational element, right? So we have chosen to use the approach where you go in and identify the components because that also brings you a bit closer to the data and allow you to do some sanity checks. But eventually we might do the more automatic approach. But I would say even when doing that, and if you want to do sort of a black box approach for the data analysis, it is extremely important to have some sanity checks and outputs that really allows you to verify that what the automatic algorithms are doing are indeed correct.
How does your pipeline handle magnetometers and gradiometers? Are they analyzed separately, or is it possible to combine them, e.g. for source analysis, (data whitening)?
Yeah, so what we are making use of is the spatial pre-widening. So we show how to do that in NME Python, but also in field trip. So that sort of brings the magnetometers and gradiometers on the same setting, if you will, and that allows you to integrate them in source modeling. So yes, we allow for combining those, so you can use the full power of the different sensor types. And by the way, it should also be possible to combine this with EEG source modeling, but that's not something we have tested yet.
May I ask why your low pass filter is set to 40 Hz before ICA? Is it because ocular and heartbeat artifact be sure to be detected under 40 Hz, and discarding information higher than 40 Hz will not lose anything?
Yeah, so I can just give a brief answer. So yes, typically you keep all the information associated with heartbeats and ocular artifacts. So what you wanted to get out before doing the ICA was the muscle artifacts, and it just due to the empirical observation that muscle artifacts are not that spatially stable, so they don't show up as reliable components in the ICA, albeit they do sort of prevent the ICA from working properly. So just by experience, these filter settings seem to work quite well when doing the ICA. But it's also very important to keep in mind that after we have the ICA components and make the projections, we apply them to the broadband filter data, right? So after you have done the ICA artifact reduction, we are still back to having the data from being at 1 kilohertz, and I think from about 0 to 300 bandpass.
Yeah, if I can add one thing just for the muscle artifact. So we suggest to use the artifact annotation in MEG Python, because as I said, it's difficult to exclude this kind of artifact using ICA. So what we do, we use an automatic method developed by the MEG people, in which by looking at the activity between 110 and 104 Hertz, which is the activity generated by the muscle, so then you can extract the power for the whole recording session, and by setting the threshold and by normalizing the data, you can then annotate all the segment of data in which the power in the muscle artifact band is higher, is too high, and that part of the data then can be removed from the analysis, for instance, rejecting epochs containing annotation with muscle artifacts.
So another question is, I haven't seen any recommendations for removing power line noise at 50/60 Hz. Does FLUX have a standardized way to achieve this?
Yeah, so already the SSS or Maxwell algorithm is going to reduce the 50 Hertz somehow, but after that it becomes sort of depending on your further analysis steps, whether you want to say use a notch filter to removing the 50 Hertz or 60 Hertz noise. So the problem being that if you apply a notch filter, yes, you reduce the 50 Hertz noise, but you also reduce everything else there, right? So you might have true brain activity in a gamma band overlapping with the 50 Hertz band. So typically what I would recommend is not to do notch filter, but then compare conditions, as also Oscar showed. And often the 50 Hertz artifacts are comparable between different conditions. So when you do the difference between conditions, the 50 Hertz is typically reduced. But if not sufficient, then you might want to do a notch filter. But be careful, because it's a double-edged sword.
So another question is, I noticed that in pipeline there is no head movement compensation included in max filter step. Any specific reason for that? Whether you would recommend recording CHPI and use it in max filter?
Oscar, you want to address this? I know we had a lot of discussions
it's a
on that.
complicated question, because there are advantages by using the head moment compensation. Because, for instance, so for those who are not familiar with it, so with this CHPI, you can record the position of the head all along the recording session. And with MEG is important because the MEG system is stable, but participants can move slightly. Maybe if they are tired, they start yawning, and then something happens. But and then if you have this information, you can use it to compensate for these moments. And it's also possible to do a temporal spatial filter during the max filter reduction. But the reason why we didn't decide to use it is because having this continuous recording of the head position means having a set of coils on the head of the participant that are always on, sending some signals, producing some signals at specific frequencies. And based on how you place these sensors, they can also interact in a non-linear way, so introducing extra noise. So we went to a more conservative, we decided to use a more conservative approach. But of course, there are cases in which it's very important to record this information, like with some patients or with kids, for instance. So in those cases, of course, it's a good idea to use it.
Yeah. So I think there's also something we would like to test more. So what we have put in the FLUX pipeline is I think we are really certain about, and we have tested them well also in the context of the Computate using a large number of data sets. So it could be after we gain more experience with the TSSS, we'll include that in the pipeline, but we are just not confident yet, simply because we haven't tried hard enough to.
So I think we have time for one last question. This is one that's been asked us. Perhaps a more general question, the exact parameters for each pre-processing step might need to change between each MEG site and perhaps each experiment. Does this mean each MEG site/experiment needs to develop its own variant of the FLUX pipeline?
Yeah, so that's a broader question, right? So of course we don't mean to be restrictive. So this is just guidelines for what worked for us, and if other people end up with slightly different parameters, that's all well and good, right? But I think it's still useful to have sort of a starting point, and then if you deviate from the starting point, you can make that explicit. So however, I would think when it comes to different sites, I don't suspect that the parameters would be site specific. So I'm not so concerned about that, but of course that could be other reasons to choose different parameters. And I should also mention this pipeline will evolve, right? So it could be that with time, some of the parameters would be adjusted and updated. But that's something we'll keep up to date on the website, and also justify when changes happens, right? But again, I mean, we are not necessarily sort of the big authority of the MEG field as a whole, right? So this is just to put forward what we are doing and what worked for us, and then other people can embrace this if they feel like it.
So I'd just like to add today, this has been a very popular Q&A session. Is there a way that people can get in touch via the FLUX website to ask further questions? I was wondering about the FLUX website, please.
Yeah, of course, people can contact me. Ole is more busy, so but I can filter things and then bring the most important things to Ole. So the codes are on GitHub. So if you're familiar with GitHub, it's also possible to ask questions and report issues, for instance, there. And I don't know, Ole, if you have anything else to add.
Yeah, at some point we might set up a discussion forum, but we are not there yet. So we're just trying to get some more resources in terms of woman or manpower to get that up running. But at some point we hope to have a discussion forum.
Well, I'd like to say thank you to both of our presenters for their time today. This has been a fantastic session, and thanks everyone who's attended the masterclass today, and we hope to see you on future masterclasses again. Thank you. Bye.
Bye-bye.